All right, so it's 3 o'clock, and welcome everyone to this interactive webinar. Is there a link between genetics and suicide? Organized by the Center for Psychiatry Research, Karolinska Institutet, and Region Stockholm. My name is John Wallert, and I'm an Assistant Professor at Karolinska Institutet, and I will moderate today's webinar. The webinar will focus on the role of genetics and suicide, what this means, and the insights genetic research can offer. With me, I also have today's lecturer and panel members, Niamh Mullins, Assistant Professor at the Icahn School of Medicine at Mount Sinai, Hilary Kuhn, Professor, University of Utah, and Manne Sjöstrand at Karolinska Institutet. This is our second public Karolinska Suicide webinar, and it will be split up in two parts. The first part is a lecture by Niamh, and part two, a panel debate together with Manne Sjöstrand and Hilary Kuhn. Before we get started, though, we have some guidelines for the webinar. Since it's a Zoom webinar, this means that you as participant can neither be seen nor heard, and you can only see the lecture presentation on your screen. We please ask you not to press the raise hand function. Instead, use the Q&A function if you have any questions. Click on the Q&A icon in the toolbar and enter your question. We will try to answer your questions as best as we can, depending on time. So without further ado, I'm delighted to present Dr. Niamh Mullins. She's an assistant professor at Psychiatric Genomics at the Icahn School of Medicine at Mount Sinai in New York. She's a member of the Department of Psychiatry, Genetics and Genomic Sciences, and the Charles Bronfman Institute for Personalized Medicine. Her lab conducts large-scale computational genomic studies of psychiatric disorders with a focus on bipolar disorder and suicide. She received her PhD in statistical genetics from King's College London, and she's both a co-founder and a co-chair of the suicide working group of the Psychiatric Genomics Consortium. Welcome.
Thank you very much, John, for the introduction, and thank you to the organizers for inviting me to give this webinar today, and to all of you out there who have joined in. So I'm just going to go ahead and share my slides. Great. So this is an outline of the webinar today. I'll start with a primer on genetics and environment to set the scene for the talk. And then I'll review the literature on family, twin, and adoption studies of suicide. I'll discuss genetic association studies to identify genetic risk variants. And finally, I'll talk about genetic risk prediction. So will this be possible? How would it work? And what are the associated ethical considerations? So first, what is heritability? Heritability is defined as the proportion of variation in a trait that's explained by inherited genetic variants. And this ranges from 0 to 100%, from genetics explains nothing about the trait to genetics explains everything. And here are some examples along that range. So the language spoken is 0% heritable. It's completely driven by the environment that a person is raised in. BMI is 40% heritable and height 80%, but still neither of those are 100% genetic. And then blood type is entirely driven by genetics. And so most human traits and diseases have a heritability somewhere in between that 0 and 100% range. And so traits like hair color, personality traits, diseases like cardiovascular disease. And heritability is a property of the population, not the individual. So it represents the average genetic contribution to a trait in the population. And everyone has a unique combination of genetic and environmental risk factors. And so heritability cannot explain why a specific person has a trait. And it also doesn't measure our ability to affect the trait. So just because something is highly heritable, it doesn't mean that it's less amenable to being altered. So for example, hair color is highly heritable. Of course, you can dye your hair. We know that diet and exercise can have an impact on BMI. And heritability is not fate. So if a trait is heritable and it exists in your parents, it doesn't mean you're destined to have the trait. It might be more likely, but it's definitely not inevitable. So most human traits and diseases are driven by a combination of genetic and environmental risk factors, including psychiatric disorders. And this JAR model for visualizing this has been developed by Dr. Janine Austin, who is a psychiatric genetics researcher and a genetic counselor. And so in the top row here, we have 4 individuals. They have different levels of genetic vulnerability to psychiatric disorders, and this is fixed at conception. It remains constant throughout life. And then over time, people start to accumulate environmental risk factors that increase vulnerability to a psychiatric disorder. And if you accumulate enough of the environmental risk factors, this jar becomes full and a psychiatric disorder may onset. But you can also see in the last row here, you can add rings to the top of the jar to prevent it from becoming full. So these would be protective factors such as social support, diet and sleep, exercise and treatment. So basically no one is born with a full jar, no one is destined to become affected, but we all have different levels of genetic risk as well as different levels of environmental risk and protective factors, and these can be modified. So we know that there are many risk factors for suicide, and here I'm grouping them by more internal and external factors. We know that psychiatric disorders are major risk factors, especially depression, bipolar, schizophrenia, also substance abuse or dependence. And personality traits like impulsivity and aggression have been associated with higher risks, as well as psychological factors like hopelessness or cognitive factors that may affect decision-making or problem solving. Early life adversity, for example, physical and sexual abuse has been linked to increased risk of psychiatric disorders and suicide in adulthood, as well as the stressful life events that occur during adulthood, things like relationship difficulties, financial difficulties, isolation or loneliness, and access to means are also established risk factors. So the question that I'll be addressing today is whether there's a genetic predisposition to suicide. And it's well known that the psychiatric disorders are heritable to varying degrees, but I'll review the evidence on whether there's a genetic predisposition to suicide specifically, and whether that's different from the genetic predisposition to psychiatric disorders. So family, twin, and adoption studies are the starting point for looking at this. They compare similarities between individuals with different levels of genetic relatedness and different levels of environmental sharing in order to figure out the magnitude of genetic and environmental influences on a trait. And this is done without any DNA samples, just by studying the genetic and environmental sharing structure. So here's an example pedigree from a family study where the colored shapes represent individuals affected with a trait through 3 generations. And family studies analyze this pattern of inheritance to calculate recurrence risk or the chance that a trait will occur again in another family member. And one caveat about family studies is that because the family members also share the family environment, this study design cannot fully disentangle between genetic and environmental reasons for transmission. And there have been many family studies of suicide, and this pedigree is from one in an Amish community where several family members over 5 generations had died by suicide. That's denoted by the S in the pedigree, and the colored shades indicate that many members of this family were also affected by mood disorders. And so reviewing the studies like this, there's consistent evidence that suicide and suicide attempt aggregate in families. And some studies also adjusted for the presence of psychiatric disorders statistically and still found aggregation of suicide within families after that statistical adjustment. But to put some numbers on that, studies generally find rates of suicide are 4 to 5 times higher in the first degree relatives of a person who died by suicide. And while this might sound like a high risk, if we consider that the prevalence of suicide in the population is about 0.02%, this equates to an absolute risk of about 0.1% for suicide. So it's actually still vastly more likely that suicide will not recur even if you have an affected first degree family member. And the family studies have generally found that the recurrence risk is higher if there are more affected members in the family. So since this study design cannot fully disentangle between the genetic and environmental causes for transmission, this is the advantage of doing twin studies, which are a powerful experiment in genetics. So identical twins share 100% of their DNA, and non-identical or fraternal twins share about 50%, which is the same as any two siblings. And twins born and reared together will share the environment within each twin pair. So this means that if identical twins are more similar to each other than non-identical twins are, this must be due to genetics rather than the environment. And this formula shown here can be used to calculate the heritability of a trait based on the observed data. And just for reference, these are heritability estimates for various psychiatric disorders from twin studies, shown here in the pink bars. So for bipolar disorder, autism, schizophrenia, and ADHD, heritability estimates are up around 75 to 80% for these disorders, whereas on the lower end, depression and anxiety are at about 30 to 40% heritability, and these disorders have greater influence from environmental factors. So a 2007 paper did a systematic review of 32 twin studies on suicide. And here they consider suicidal ideation, suicide attempt, and suicide together. So I'll refer to that broadly as suicidality. And heritability in these twin studies has ranged from 30 to 55%. So that's something on par with depression or anxiety. But some twin studies also adjusted statistically for the presence of psychiatric diagnoses, and heritability after that adjustment was between 17 to 36%. And they've shown that the personal environment accounts for almost all of the remaining risk, and the family environment has little, if any, role to play in suicidality. And from other twin studies, there are not many large studies for suicidal ideation, suicide attempt, or suicide separately, nor estimates in males and females or at different age groups. And this is because the data are just too sparse to get reliable estimates in these groups. So in case you're still not convinced that this is not being driven by shared environment within twins or within families, another study design which further reduces potential environmental sharing is an adoption study. And adoption studies compare similarity between an adopted individual and their biological parents versus their adoptive parents. And so, of course, they share only genetics with the biological parents and only environment with the adoptive parents. And the classic adoption study on suicide was published by Schulzinger et al. Using the Danish Adoption Registry. And so they studied 57 adopted individuals who died by suicide and found a 6-fold higher rate of suicide in their biological relatives versus their adoptive families. And this was regardless of whether the adopted individual had a psychiatric disorder or not. And so this again supports the role of genetics and not the family environment. And there have been similar findings from other studies in other Scandinavian registries. So there's convergent evidence from these studies that suicidality is moderately heritable, and it's not explained by the genetics of psychiatric disorders alone. But these kind of studies don't give any information about which particular genes or genetic variants are involved in suicidality. And so this requires collecting and analyzing DNA samples and doing genetic association studies. So I'll just first give a quick primer on DNA variation. The human genome is about 3 billion base pairs, and it's 99.9% the same in all humans. The remaining 0.1% comprises genetic variation that makes individuals different from one another, and there's various types of this genetic variation. The most common is the single nucleotide polymorphism, or SNP. And let's say this is the DNA sequence of two individuals. You can see it's mostly the same in the sequence, but at this one highlighted position, one individual has a C allele and the other individual has a T allele. Insertion deletion variants, or indels, occur when one or more base pairs are present in some sequences but absent in others. And then copy number variants occur when there is a repetition of identical or nearly identical sequences in some genomes. So we're all carrying these kind of genetic differences, and these differences can be associated with increased likelihood of having a certain trait or an illness. But we typically refer to them as genetic variants rather than genetic mutations. So I think a mutation suggests something that's pathogenic or atypical, but the majority of these variants are not pathogenic by themselves. And there's many types of genetic association study depending on the type of genetic variation you're looking at and the technology that's used to measure it. In the past, a lot of candidate gene studies were done in psychiatry, which involved genotyping or sequencing a handful of genes based on some prior hypothesis that they were involved in a disorder. And this kind of work generated a lot of false positives and non-replications in the field. So these kind of studies are not really done anymore. But in contrast, genome-wide association studies involve genotyping common SNPs across the entire genome with no prior hypothesis about which genes are involved. And there have been many large GWAS done in psychiatry with very successful results. And it's now about $40 per genome to generate this kind of data. So GWAS only covers common genetic variation, but whole exome sequencing sequences the coding regions of the genome, and it has the advantage of being able to detect rare variants. And finally, whole genome sequencing does exactly what it says. It detects everything. And with recent technology, just in the last couple of months, the price of this has dropped to about $200 per genome. So this will enable many more whole genome sequencing studies in the near future. And in suicide research, there has been some whole exome and whole genome sequencing studies done, but these are still small. And the area where there has been most activity is in GWAS. And so this is the area that I will focus on today. So this is the pipeline of a GWAS. We start out with ideally a large sample of individuals, a group of cases who have the trait of interest, as well as controls who don't have the trait. And DNA is collected from these individuals and genotyped on a microarray. And this technology has probes which can detect which allele is present at SNPs across the genome. And after quality control of the data, we examine genetic ancestry of study participants by comparing them against established reference panels from different world populations. And this is because it's crucial to make sure that the cases and controls in the GWAS are well matched to each other in terms of ancestry. And then in the analysis step, we compare the statistical frequency of alleles between the cases and controls in the study. And when an allele is at a statistically higher frequency in cases compared with controls, it's said to be associated with the trait. And we do multiple testing correction for 1 million SNPs, which is the estimated number of independent SNPs across the genome. And it's also very easy to meta-analyze GWAS results across studies to further increase sample size and increase the statistical power. And so the results are typically displayed in a figure like this. It's called a Manhattan plot. And on the x-axis, we have the SNPs analyzed across chromosomes 1 to 22 in the study. And then the y-axis shows the minus log 10 p-value. So the higher the peak, the more significant the association. And so this may detect novel genetic risk loci for the trait of interest. But besides that, the results can also be used to do many types of follow-up analysis. So they can be integrated with other types of genetic data to gain insights into biological pathways, drug targets, genetic overlap with other traits or disorders. And there have been many GWAS of suicide over the last 10 to 15 years. And here I'm breaking them down by GWAS of suicide attempt, suicidal ideation, and suicide, as well as some studies on severity of attempt or suicidality as a spectrum. And you can see that the number of cases in these studies have dramatically increased over the years. And I will show the results from some of the 3 latest studies with the focus on suicide attempt. So since extremely large sample sizes are required for these kind of studies, no single research lab could tackle doing this alone. So in human genetics, there are many consortia of scientists worldwide who combine GWAS data through meta-analysis to do powerful GWAS studies. And we recognize the need for this approach in suicide genomics to really drive this field forward. And so in 2018, we established a consortium for doing this. We are the suicide working group of the Psychiatric Genomics Consortium. And the PGC also has work groups for all of the major psychiatric disorders doing similar genetic research. And our goals are to uncover the genetic and biological etiology of suicide attempt, suicide, and suicidal ideation. And I co-chair this group along with 2 of my colleagues, Anna Docherty and Doug Rudifer. But the group is made up of over 260 members from all over the world. It's a multidisciplinary team of geneticists, psychiatrists, ethicists. It takes a huge team and a multidisciplinary group to do this kind of study. And the figure here shows all of the cohorts that have participated in our GWAS to date. And at these links, you can find us on the PGC website to get more information about the group. So I'll tell you a bit about the design of our GWAS of suicide attempt. And we have 1 paper published on this, but we have a preprint currently on medRxiv by Docherty et al. And so I'll share results today from this newer study, which is currently under review. So there's 22 cohorts in the study. Most of these were established for doing genetic studies of psychiatric disorders like depression, bipolar disorder, schizophrenia. The participants also have information available on history of suicide attempt through their psychiatric interviews. A couple of studies were ascertained specifically for research on suicide attempt or suicide, and 6 of them are from population biobanks. 80% of individuals in the case group in the study had a history of a lifetime suicide attempt. So that's defined as a non-fatal act of deliberate self-harm where there was intent to result in death. And the information on that is usually from psychiatric interviews with clinician or a trained researcher, but we had 2 studies that used self-report and a few studies that used hospital records like ICD codes or reason for admission. And then the remaining 20% of the cases in the study were individuals who died by suicide, and these DNA samples have been collected through collaboration with coroners' offices. The controls can best be described as a general population sample. So they're screened for the absence of suicide attempt if possible, but they're not screened for the absence of psychiatric disorders. And we have a total of over 43,000 individuals in the case group and over 900,000 individuals in the control group. The majority of the study are participants of European ancestries, and this is very typical in GWAS currently, but we do have some representation from African American, East Asian and Latinx ancestry groups. So we've done this by doing a GWAS in each of these studies within each ancestry group, and then doing a meta-analysis across all of the cohorts to achieve the final sample size. And this is the Manhattan plot of results from the full meta-analysis. So as a reminder, the x-axis shows every SNP analyzed, and the y-axis shows the -log10 p-value. And the red line here represents the genome-wide significance threshold of 5 × 10⁻⁸. So that's after that multiple test and correction for 1 million tests across the genome. And we found 8 genome-wide significant risk loci. So that's 8 regions of the genome where alleles occur at a statistically higher frequency amongst individuals with a history of suicide attempt versus the control group. But this doesn't mean 8 risk genes for suicide attempt. And if we zoom into one of these risk loci, the Manhattan plot looks like this. And so there can be multiple genes within these regions, and we don't necessarily know from this work alone which is the biologically relevant gene, and we have to do further types of post-GWAS analysis to disentangle that. And so the genes that are on this plot here are just the nearest gene to the most significant SNP in the locus, not necessarily the causal gene. And I will show one of these loci in more detail. So that's the intergenic locus on chromosome 7, which is the most significant risk locus identified by the GWAS. On the left here, I'm showing the regional association plot, so zooming into that risk locus. And you can see here that the significant SNPs are intergenic, and there are multiple genes nearby in this region. These significant SNPs have not been linked to any of these particular genes. And on the right is the forest plot, which shows the effect size for the most significant SNP at the locus on suicide attempt. And interestingly, it's also been associated with increased risk of insomnia, smoking, and risk-taking behavior from other large published GWAS. But it hasn't been associated with any psychiatric disorders from GWAS, indicating its specificity for suicide attempts. And we have also done sensitivity analysis by conditioning the GWAS results on the genetics of depression and other psychiatric disorders. And this association with suicide attempt remains, indicating that it's not just driven by the genetics of depression. And so the allele at this locus is associated with a 1.05-fold increased risk for suicide attempt, and also increased risk for insomnia, smoking, and risk-taking behavior, but not psychiatric disorders. And since the relevant gene or mechanism here is currently unclear, we have further work ongoing within the group to dissect that using various types of data. There's 2 other important properties of the GWAS of suicide attempt that I want to highlight, which give us information about the genetic architecture of suicide attempt. SNP heritability is the proportion of variation in suicide attempts that's explained by SNPs in the GWAS. So that's part of the overall heritability of suicide attempt estimated by twin studies. And there's software tools we can use to estimate this. And we have calculated that the SNP heritability of suicide attempt from our GWAS data is 6.8%. And after we use that statistical conditioning approach that I mentioned to condition the GWAS of suicide attempt on genetics of depression and other psychiatric disorders, we saw that SNP heritability estimate decreased to 4.1%. So this 4.1% can be interpreted as the SNP heritability of suicide attempts that's independent of the genetics of other psychiatric disorders. And just as a reminder, I'm putting the twin heritability estimates here for suicidality, both before and after conditioning on the genetics of psychiatric disorders. So the big difference between the SNP heritability and the twin heritability is those other types of variation that we're not studying in the GWAS, so like copy number variants or rare variants. But what I wanted to highlight here is that both the twin studies and GWAS indicate that there are genetic contributions to suicide attempts that are not driven by the genetics of depression and other psychiatric disorders. And then the second property is polygenicity, which refers to the number of genetic variants that influence suicide attempt. And so while we found 8 genome-wide significant risk loci from this GWAS, there will also be many more genetic associations with suicide attempts that we expect to find as sample sizes increase. And we used a statistical method to estimate the number of those genetic risk loci that exist. And this estimated 8,300 independent SNPs that account for that observed SNP heritability of suicide attempt in our data. So the take-home point here is that there is no single risk gene for suicide attempt, but rather there's thousands of genetic variants across the genome that each contribute a small increase in risk. And if you inherit many of these genetic risk variants, then genetic risk can start to accumulate. Once we have the GWAS results, there's many types of follow-up analysis, as I said, that we can do with these by integrating other types of genetic data to gain insights into underlying biology. So I just want to give a couple of examples of the type that work that we're doing in this area. So on the left, I'm showing results of a tissue expression enrichment analysis. So here we take GWAS results and we can ask, is there an enrichment of genetic associations with suicide attempts in genes that are known to be expressed in various tissues beyond what would be expected by chance? And so we have done this using data from 54 bodily tissues from the GTEx Consortium. And we see a significant enrichment of genetic associations in genes expressed in all of the brain tissues and no other tissues in the body. And we can also do similar analysis to this using particular cell types of the brain, which is a future direction for us. On the right, I'm showing the results of an SMR analysis. So this is where we're integrating the GWAS results with data on EQTLs from brain tissue. And so EQTLs are alleles that influence gene expression in the brain. And by integrating these two data types, here we find evidence to suggest that the alleles that increase risk of suicide attempt at the chromosome 15 genome-wide significant locus act through increasing expression of the FES gene in the cortex of the brain. And so FES is encodes a protein with tyrosine kinase activity. It's involved in a range of cellular processes like differentiation and cellular migration. A topic of strong interest to us is using genetics to identify potential opportunities for developing drug treatments for suicidality that are safe and effective in the diverse group of individuals who are affected. And so these are the results of a drug-targeted enrichment analysis. And this is just like the tissue expression enrichment analysis. But here, instead of genes expressed in a tissue, we're looking at genes that are targeted by different classes of drugs. And the drug classes and the gene targets we obtain from online curated databases. And here we examine 63 drug classes. We see significant enrichment of genetic associations with suicide attempt in genes that are targeted by antipsychotics, including mood stabilizers, psychoanaleptics, as well as some particular classes of antidepressants. And as the size and the power of our GWAS increases, I anticipate we'll see enrichment in more classes of drugs, and we can also start to do this kind of analysis for individual medications. And so this can pinpoint potential opportunities for repurposing some existing classes of medication for the treatment of suicidality. Here I'm showing genetic correlations of suicide attempts with some human traits and disorders. So pleiotropy is when genetic variants affect more than one trait, and this is widespread. And it can be easily measured using GWAS summary statistics or results by calculating genetic correlation, which is the correlation between the genetic effect sizes on two different traits from GWAS. And this ranges from -1 to +1. So -1 meaning that all genetic variants have the opposite effect on two different traits, and +1 meaning that they have the same effect. And so here you can see that the strongest genetic correlation with suicide attempt is self-harm behaviors, which also includes non-suicidal self-injurious behavior. And we have repeatedly seen that major depression is the psychiatric disorder that shows the strongest genetic correlation with suicide attempt, with an overlap of 0.68. So that's a strong genetic correlation, but it's still much less than 1, so they're not genetically the same. But we see positive genetic correlation with all psychiatric disorders, as well as I'm noting here chronic pain and risk tolerance, because these have been shown to be associated with suicide attempt on the phenotypic level. So these results suggest some shared underlying biology between suicide attempt and these disorders or traits. And so while I focused on GWAS of suicide attempt so far, there's also GWAS underway of suicidal ideation and suicide specifically. And so in the largest GWAS of suicidal ideation to date, which has been conducted in the Million Veteran Program Biobank in the US, there were over 99,000 individuals in this biobank who had evidence of suicidal ideation. And this study found 5 genome-wide significant risk loci. In terms of GWAS of suicide, the largest study has been done at the University of Utah, where a research team have been collecting DNA samples through a collaboration with the Office of the Medical Examiner for over 10 years. And so this study found 2 genome-wide significant loci and a higher SNP heritability estimate for suicide at 16%. And so using these results, we can also ask the question, are the risk alleles for suicidal ideation, attempt, and suicide the same or different? And this can easily be calculated, again, by looking at genetic correlation between the GWAS. And so what we have seen is that there is a genetic correlation of 0.77 between suicidal ideation and suicide attempt. And also between suicide attempt and suicide, again, a correlation of 0.77. So there's strong but incomplete genetic overlap between these 3 phenotypes, and this indicates that they're partially genetically different and therefore also likely to be partially biologically different, rather than one of them being a more severe manifestation of another. And so understanding these similarities and differences between these suicide outcomes could uncover risk factors for transition from one to the next. And this is something that we are planning to study in future work in the PGC Suicide Working Group. So we're further expanding our GWAS efforts to increase the number of cohorts included and to study the genetics of suicidal ideation, attempt, and suicide separately. And these are our projected numbers in the next 5 years, and we continue to do global outreach to scale this up. And we know from GWAS of other psychiatric disorders that increasing sample sizes increases genetic discovery, and our goal is to use these data to find novel risk loci, gain insights into relevant cell types, pathways, drug targets, and to enable studies of potential modifiable causal risk factors for suicide as well. So one topic I was asked to touch on today in the talk is evolutionary theory on why these risk alleles for suicide or for psychiatric disorders persist if they're associated with early mortality and morbidity, and why are they not removed from the gene pool. And there's a whole field of evolutionary genetics and evolutionary psychiatry, and these are some of the main evolutionary theories that have been proposed to explain this. So the mismatch hypothesis is the idea that these alleles were advantageous in the past to our ancestors, and only recently have they become associated with mortality and morbidity in the current environment that we live in. Balancing selection is the idea that these alleles are advantageous to some individuals, for example, up to a certain threshold, and this compensates for the negative or disadvantageous effects in other individuals. And I have Vincent van Gogh up here because one of the most common balancing selection theories in psychiatry is that risk alleles are associated with increased creativity. And then polygenic mutation selection balance says that these risk variants are under negative selection pressure, and they are being removed from the gene pool at a rate that's proportional to their effect size. And so rare variants with large effects will be removed faster than the common SNPs with small effects that we're looking at in the GWAS. But new mutations or a new genetic variation arises all the time. And for brain disorders, since they're so polygenic and half of all genes are expressed in the brain, there's a large scope for a new variant to have an effect on brain function. And so there have been reviews and theoretical modeling papers published on these evolutionary theories in psychiatry, and they have discredited the mismatch or balancing selection hypotheses as explanations, and they've not really stood up to the mathematical calculations to explain this. And so the most likely reason for their persistence of these alleles is the polygenic mutation selection balance theory. So just to summarize the genetic association studies, there have been large GWAS of suicide attempt, ideation, and suicide, which have identified risk loci. There's no single risk gene, but there's many alleles with small effect sizes on risk, and integrating GWAS with other kinds of biological data can provide novel insights. And in the final section, I'll now discuss genetic risk prediction. So what is this, and would it be possible to do this for suicide? For a polygenic trait, a single variant is not informative for predicting risk, and instead, typically a polygenic risk score is used. And this is a weighted sum of the number of risk alleles an individual carries with those alleles and their weights obtained from large genetic association studies. And a PRS is a continuous score per individual per trait, and all individuals are carrying DNA risk variants for all common traits and diseases. So if you plot a PRS score, as I've shown here, it looks like a normal distribution in the population. And these are pretty simple to calculate, they're inexpensive, and they're fixed at conception. And so the figure on the right shows the potential stages where polygenic risk scores could be used in the clinic throughout life, from doing risk prediction at birth to clinical assessment in the early stages of an illness, assisting with differential diagnosis, maybe selecting treatments, and to predicting the course of an illness. But there are some important caveats to keep in mind with PRS. So one is that the accuracy of the PRS depends on the identification of those risk alleles and their appropriate weights from the genetic association studies. So their power will be driven by the research that we're conducting. And then the second thing is that the heritability of the trait is the maximum variance that a genetic risk predictor could ever explain. So for suicidality, this would be in the range of 30 to 55%, but that's only if we knew absolutely everything about all of the genetic effects on suicidality, including rare variants, copy number variants. And in reality, what we're actually studying and most often using is GWAS data, those common genetic variants with small effect sizes. So in using those types of variants to calculate a polygenic risk score, their upper limit of performance is SNP heritability of the trait, which for suicidality is more like 6 to 16% from what we've seen so far. And so basically on their own, PRS will never be able to definitively predict a future suicide attempt or suicide because genetic factors only contribute part of the risk. But that doesn't mean that they can't provide some clinically useful information. And this is a PRS workflow, just to briefly explain how it works. So we take the results of a large, well-powered GWAS, ideally much larger than we've been able to do to date. And we use this to select genetic variants and their weights, and then calculate the PRS for each individual in some validation data sets, where the information on history of suicide attempt is known to us. And then we evaluate the PRS using known case control status in these data sets to see how well it performs. And so this figure here is real data on PRS for suicide attempt from our GWAS, showing the variance it explains in 6 independent validation data sets. And current PRS for suicide attempt explain about 1% of variance in suicide attempt versus control status. So obviously that leaves 99% of the variance unexplained. And as our GWAS increase in sample size, we will be able to do a better job of this. We'll be able to increase that percentage, but still its upper limit is SNP heritability. And then in step 4, what I'm showing here is what we would ultimately like to be able to do, which is to calculate PRS for individuals with unknown status and then benchmark them against the population and say, okay, this person is in the top 10% of PRS or the top 1% of PRS, and their risk is increased by a certain amount, and be able to use that information along with other predictors to make some kind of decision on clinical management. And so while this is not currently clinically useful for predicting suicide, I'd like to discuss a bit about the future potential as the power of these PRS improves. So we can look at schizophrenia, which is the psychiatric disorder that we know most about genetically. And current PRS for schizophrenia explain about 7% of variance in schizophrenia versus control status. And so this is insufficient for predicting schizophrenia in the population, but research has shown that there are significant differences in PRS for schizophrenia between patients with first episode psychosis who go on to receive a diagnosis of schizophrenia versus another psychiatric diagnosis. So in this way, the PRS for schizophrenia could be useful for providing information to inform a clinical care plan for these individuals. Also, for most people affected with schizophrenia or a psychiatric disorder or suicide, they don't have a family history. Given the low prevalence of these disorders in the population. So a family, a polygenic risk score can provide information that a family history cannot. And there are also no clinical biomarkers to exist with this. So from that perspective, the PRS is an objective measure that's based on biology. So if we look to the future, I think we can assume that genetic risk prediction will improve, but clinical use of PRS in psychiatry, I think, is unlikely in the short term. And we should look at these as a possible addition, but not a replacement for other risk predictors, given what we know about their maximum levels of performance. And there are some views in the field about the likely long-term use of PRS in psychiatry. being stratification of high-risk individuals who might benefit from in-depth assessment to target early interventions to improve outcomes, maybe motivating lifestyle changes in high-risk individuals, for example, avoiding recreational drug use if you know you have a high PRS for schizophrenia. Or in guiding clinical care for patients who present with general symptoms, and to identify groups that are more likely to go on to develop a severe psychiatric diagnosis. But having said that, of course, translating this to the clinic comes with many ethical considerations, particularly for suicide. And there has been a recent paper on ethical considerations related to genetic risk prediction for suicide. And the authors argue that for the foreseeable future, the potential for harm to individuals from marketing a genetic risk score for suicide is much greater than any conceivable clinical benefit. And I agree with this view at present, but I think that even in the future, when these PRS improve and potentially are clinically useful, many of the ethical issues that are raised in this paper will still be relevant. So, for example, we need improved genetic risk literacy amongst the public and clinicians, and training of clinicians to communicate this kind of information. Genetic determinism is the false belief that genetics is destiny, and this kind of misinterpretation of a PRS could have damaging consequences, like creating a sense of hopelessness. It could reduce motivation to seek treatment, reduce confidence in treatment. A major issue is that currently the accuracy of PRS are much lower in individuals of non-European ancestries. And so this has the potential to exacerbate healthcare disparities between populations. Genetic testing also impacts family members and family relationships. It may be associated with stigma or discrimination. Children or some vulnerable adults may have reduced ability to comprehend and to use the information from genetic testing. And there should be an actionable consequence of genetic testing, as well as availability of resources to action. And we know that resources are limited in many places. But on the flip side, there are also potential positive consequences. For example, knowing information on genetic risk might make individuals more cognizant of early symptoms and encourage early treatment seeking, which will lead to improved outcomes. It may promote protective behaviors, and it could also reduce self-blame and shame for individuals who are affected. And of course, the possibility of being able to use this to direct interventions to those in need and to areas where these interventions could have the greatest impact would be a huge advantage. So this is quite a new field, and we will need careful clinical studies of the impact of doing genetic testing in psychiatry. The relationship between genetic testing and mental health could be very complex. So I hope that will leave you with some food for thought on this topic. And before I wrap up, I'll just briefly recap on some of the take-home points from the webinar. So we have seen convergent evidence from family, twin, adoption, and molecular genetic studies that suicidal ideation, attempt, and suicide are moderately heritable, and they're not just explained by the genetics of depression or other psychiatric disorders. They're highly polygenic, so there's no single risk gene, but there are thousands of genetic variants with small effects on risk. And because of this, we need huge studies to detect these genetic variants, but such studies are possible. Integrating results from these studies with other types of biological data can yield insights into relevant pathways, cell types, drug targets. And genetic risk predictors may in the future be added to current risk prediction models, although not replace them. And this is unlikely to be in the short term. And then finally, I think the most important takeaway is that genetics is not fate. It only alters risk. We have many unique combinations of genetic and environmental risk factors, and risk can be reduced with proper support and treatment. So with that, I'd like to thank the members of the PGC Suicide Working Group. It takes a large and diverse team to do this kind of research. And I'd also like to acknowledge all of the participants in these studies. They've shared their DNA and their life experiences for this scientific research. And so without them, this work would not be possible. And thank you to everyone again who's joined the webinar. And I will hand it back over to John.
Thank you, Niamh, for a very nice presentation. Now we're entering part 2 of the seminar, where there will be a panel debate. But first, I thought I could check if there are any questions from the audience. I could pose one myself. See, I don't think there are any questions. I was thinking of the you mentioned the GWASs and based on common variants. Is there work ongoing on rare variants and copy number variants to be combined for prediction, or know that?
Yes, there are some studies that have generated whole genome sequence data and some exome sequence data. And with recent reductions in cost of whole genome sequencing, I think we'll see a lot more whole genome sequencing studies in the near future. And so, yes, rare variants could be combined with common variants to do genetic risk prediction, and that's something that would definitely be advantageous to do. Of course, then we need these validation data sets that also have sequence data available. So I think it would take some time for that to catch up. But yes, it would be possible, and I think it would be a real strength to do that.
Nice. I was also thinking of the high sort of genetic correlation between the different phenotypes that are interrelated, like suicide attempt, suicide ideation, and suicide death. Did the SNP hits, do they also are they the same also across the different traits?
Yeah, that's a good question. So none of the genome-wide significant loci that have been found, if I remember correctly, are the same across the 3 studies so far. There are some that are, say, genome-wide significant for one, that are just below significance for another. But this is what we expect, initially with the sample size of all these studies. They won't necessarily find the same G1 risk loci first, even if they are implicated in all 3 of the phenotypes. But given the strong genetic correlations that we have seen, I think that in the future we will see more overlap of the genome-wide significant loci between the 3 phenotypes. And there could also be a rationale for meta-analyzing them in the future to further increase statistical power, or maybe even use new methods for doing meta-analysis that leverage that shared genetic component between the 3 phenotypes.
Thanks. And we got a question from the audience here. And before I let in the panelists, the question is from Mikael Landén, and he says that, he asks, the suicide attempt rate change over time differs considerably across population. Is this a problem for genetic research? And if so, how and how can it be handled?
And thanks for that question. So yes, indeed, we see different patterns in the epidemiology of suicide or suicide attempt over time. I don't think that necessarily equates to genetic differences in its etiology over time. It would be difficult to study that, I think, but in terms of differences between populations, this is definitely something that we would like to study. And so one of our major priorities is increasing ancestral diversity in our studies. So we continue to do outreach all over the world to increase the representation from different ancestry groups. And so once we're able to do that and further increase our sample sizes, we'll be able to look at genetic similarities or differences between different ancestries. Thanks.
Now there are more questions dropping in here, but I think we should move to the panel debate. And so I'm very glad to welcome Hilary Kuhn and Manny Sjöstrand. Hilary Kuhn is a renowned and highly cited professor in psychiatry and also an adjunct professor in biomedical informatics and internal medicine at the Department of Psychiatry, University of Utah. Her primary research interests include finding genes that increase susceptibility to autism, a genetic risk factor for suicide, and the genetics of nicotine and alcohol addiction. She's frequently involved in ongoing psychiatric genomics consortium work on suicide genomics. And she also works a lot with a very interesting database, the Utah Population Database on suicide. So welcome, Hilary. And we also have Manny Sjöstrand, who's a psychiatrist and ethics researcher at the Department of Learning, Informatics and Management and Ethics at Karolinska Institutet. His research interests include ethical aspects of suicide prevention and prediction in healthcare. So welcome.
Thank you.
Thanks very much.
I guess the floor is yours if you have specific questions.
Sorry, go ahead.
Go ahead, Hilary, sorry.
So I wanted to follow up a little bit on the idea of sort of subsets within this bigger group that you're studying that is, suicide attempt, such a heterogeneous condition. And one aspect of that, I was just thinking would be natural to think about a little bit is the idea that you have the very big million veteran program data set that's part of this really large data set, but it's a very big data set. And how the ascertainment in that data set is so different. So all of this work really started very much more narrowly with people, as you mentioned, who were ascertained from psychiatric clinics, who have psychiatric conditions, and then on top of that have suicidality. And then you have this big million veteran program data set and some other data sets, including ours, that are much more population ascertained and include individuals that are sort of all across the spectrum, including, a lot of people maybe that don't get a lot of psychiatric care. And just how that is a complicating factor in the way that you're thinking about risk and how it might be able to tease out some risk subsets where, like in the Million Veteran Program, maybe the underlying genetic vulnerability is sort of different, and then their exposures are different. So they are going to maybe give you a different signal from some of the people that are like from a depression clinic.
Yeah, we have actually done some work to look at that. So there has been a GWAS of suicide attempt published in Million Veteran Program specifically. And so we have looked at the genetic correlation between that GWAS and the first GWAS that we published as the International Suicide Genetics Consortium, or PGC Suicide Working Group, which comprised mainly those individuals who were ascertained based on psychiatric disorders from inpatient or outpatient psychiatric clinics. So yes, the characteristics of the individuals in these two studies are quite different. MVP are of course a veteran population. They likely have very different environmental exposures than individuals in the rest of our studies. But what we have found is a very high genetic correlation of suicide attempt between the two groups, not significantly different from one. So this was very encouraging to see that high similarity in the genetic etiology of suicide attempt between those two groups, despite their differences. Encouraging in terms of doing the meta-analysis and reassurance that the phenotype is robustly defined across the cohorts. But we do plan to look at this a bit further. For example, in MVP, there's major gender differences. It's a predominantly male sample, whereas in the rest of our studies, they're predominantly female. There could possibly be sex differences in genetic etiology, and we haven't had the chance to look at that yet, but it's a future direction with further increases in sample size.
Great, thanks very much. There's just one other thing that I could follow up on that's sort of similar, and that is thinking about, you mentioned the pretty strong correlation between suicide ideation, suicide attempt, suicide death. And in our study, which is really predominantly focused on suicide death, we see only about half the individuals who died by suicide have a prior non-fatal attempt, which is really interesting. And those people tend to match much more closely to the individuals in the bigger data set that are suicide attempters. And the people that don't have any prior suicidality are interesting and appear to be much less sort of psychiatric. So it's an interesting idea to think about those who have a sort of a history of attempt are kind of a one group. And then the aspect of this that you mentioned that was like an association with chronic pain, I was thinking maybe that's kind of starting to be a window into thinking about some of these individuals who are suicide deaths where we're really bad at predicting them. They don't have anything on the radar where they have any prior suicidality. And it's actually a lot of people, especially once we get out into ascertainment in sort of larger population ascertainment. So I think that some of this work that you're doing is so important in regards to not only disentangling all of the sort of psychiatric comorbidities and how that's working, but also in sort of getting into some of the aspects of this that are less psychiatric and more driven by other things, such as the chronic pain signal that you're starting to see.
Yeah, absolutely. I know, Hilary, in your data, you have medical records, and so you're able to ascertain information on whether individuals who died by suicide had made a prior suicide attempt. And I think, as you say, it would be very interesting to look at the genetics of those individuals who had not made a prior suicide attempt. Using your data, or another approach we could potentially take in the future is to use methods that do case GWAS, where we can look at genetic differences using large GWAS of suicide attempt versus suicide, to try and disentangle those genetic effects. But I agree that the genetic overlap with non-psychiatric phenotypes like pain will be really interesting to look at further.
And also, I guess possible intervention in the future would be quite different for those two, right? Interesting. I'm thinking of the ethics of this. I mean, it's a very interesting part, and I'm glad that this came up in your slides, Niamh, and I know that you work a lot on this. Do you have anything to add regarding that? The ethics of suicide risk and genetic risk and risk prediction?
I mean, it's a very interesting topic. It's quite a new field, as I mentioned, and so I can really only hypothesize on the potential consequences, both positive and negative, in the future. There's so many things to consider. There are, for example, some clinical trials ongoing in other disease areas, like returning PRS for breast cancer to patients, and seeing what sort of impact that has. So I think that doing these kind of studies will be really crucial in psychiatry, and especially when it comes to suicide, which is such a sensitive topic, to see what sort of impact returning this kind of information has in different contexts, and further work to improve genetic risk literacy for the public and clinicians.
So, you mentioned that currently polygenetic risk scores are not clinically useful. What do you think? When could they be, and what is missing in order for them to be clinically useful? Do you have any ideas, and what kind of research would be needed to fill that gap?
Yeah, so first of all, we need to really increase the sample size of the genetic association studies that we're doing. And what we've seen from studies of other psychiatric disorders is that as you do that, if you look at the trajectory over time, the performance of the PRS, those percentage variance explained will increase with the power of the GWAS studies. Given that their performance at the moment is poorer in individuals of non-European ancestry, that is a major consideration, and we are actively working to, as I said, increase ancestral diversity in order to improve this and make these PRS equitable for different populations. And then I think in terms of looking at their use in the clinic, we'll really need research studies to actually look at the associations of these PRS with particular clinical features in patient populations, and to see if they can discriminate between, for example, as I mentioned, in the patients with first episode psychosis who go on to receive different psychiatric diagnoses. So we'll need those similar studies for suicide, but we're just not really at that stage yet.
So one question I don't expect you to have an answer to this question, but maybe Hilary does, is, do you think, and I can ask the panel, do you think people would like to know their risk score for suicide, and or would they like to know that in the relatives? And so I'm just, what personal reflection here is that I, as a consultation liaison psychiatrist, I meet lots of patients, and one of the most common questions that I get from my colleagues in other specialties, and one of the most common reasons for referral is suicide risk, and they want me to make a suicide risk assessment of patients. But I've never, I actually have thought about this, and I'm not sure, but I don't think I've ever met a patient who has him or herself asked me, or the main reason for seeking my counsel is to ask about the suicide risk assessment. They've sought help for many other reasons, of course, and a host of reasons: anxiety, depression, etc., and also, of course, things related to suicide, but not about the suicide risk assessment. So that this is more of a general open question, and I think that, Hilary, I know that you've done research on these relatives to and the public opinions about and views on.
Yeah, so we conducted research that so far I think is too small. It would be nice to do a lot more of this. But what we did was several focus groups with people who are either survivors of suicide attempt or family members of individuals who died by suicide. So they're really stakeholders. And we asked them a lot of questions that are exactly what you're getting at, about sort of what they want to know. What do they see as the risks and benefits of knowing? How do they sort of see this playing out? How do they understand what that risk means? And it was really interesting. So for the most part, people said they would want to know. Of course, these are people who have sort of been through the experience. So I think what we need to do is paddle this back a little bit and get at groups that you're talking about where the risk is not quite so apparent yet. For the individuals we talked to, it was one really strong outcome of this was this idea that even just by studying suicidality as a genetic trait, this was reframing it as a medical condition, and that was incredibly empowering, that it was really liberating as far as sort of stigma, which I think Niamh touched on. That being said, there was concern about sort of the fatalistic interpretation of results. We've seen this, I think, in some studies where people have gotten their polygenic risk for diabetes, and the assumption is people would change their lifestyle, they would, change their diet and all this kind of thing. And there are quite a good number of people who don't do that and say, well, it's my genetic risk, there's nothing I'm going to do about it, I might as well just eat. And that's the risk for, giving out information about genetics of suicidality without the proper interpretation of that, is that individuals might just say, well, this is my genetics. And then, the worst outcome, I might as well just kill myself now, get it over with. So we really, really, really want to avoid that, this sort of deterministic model, the fatalistic issues. And then also, this is, we need to have an answer. You give somebody their risk, there needs to be a way to mitigate that risk. There need to be access to the right services. There needs to be knowledge of how that risk interacts with environment, which we're really not very good at, even if we get to the point where we can interpret better with polygenic risks. What exactly are the environmental factors and the timings of those environmental factors that really accentuate that underlying genetic risk? We don't understand that very well, and that's going to be a big challenge. So I think there's a huge benefit for these studies, and we're not going to stop them. Obviously, we need to understand what these risks are. But the way that the information gets presented to the public and to our patients, it's really going to be very, very important.
Yeah, I'm sure there will be personal preference for people, whether they would like to know or not, but also that preference may change as genetic risk literacy improves. And so, I mean, family history is already something that's very commonly assessed in medicine. And so you could think of this in the same way. And I think we will all have our genetic information increasingly available to us in the future. So it's really important to be doing the kind of studies that Hilary is describing and seeing the impact of this on patients. And I mean, it's very different to provide this information to someone who's well and is not having any symptoms compared with somebody who's already ill, and then it may be an explanation for the symptoms that they're experiencing, which could lead to less self-blame or shame about how they're affected. So I think it will be important to do those studies in different contexts.
I agree. So, thanks. Yeah. I think we got an interesting question from the audience. If it's right to pick, if you want. About,
do you mean the one with the future?
About does genetic add prediction above known clinical risk factors?
Yeah,
that's an excellent question. So Hilary and I have a project underway actually looking at this, where we are using biobank data in several hospital systems in the US, where we are calculating PRS for individuals and combining that with other clinical information to see what it adds. So that's ongoing work. And Hilary, if you have anything to add on that from other studies, please go ahead.
Yeah, I mean, I do think this is really important, and that, again, one of the things that we've started to do with this is to think about it in a much more complex way, where we're not thinking about one polygenic risk that identifies the whole set of people, but rather we're thinking about different types of or constellations of polygenic risk that pull out different subgroups of individuals at different underlying genetic risks. So there, for example, I mean, you could think about the idea that there are individuals who are very young, who are maybe at very different risk from the individuals who sort of start in on their suicidality and they're later in life. And that's a very simplistic difference. And we think there are probably constellations of underlying genetic risk that we can start to tease out by looking at these really large data sets that will get us at a better picture of probably much smaller groups of individuals who are at more specific risks, where then thinking about treatments and environmental risk factors that are magnifying those biological risks is going to be a bit more tractable.
I mean, I guess the definition of the target phenotype, that's the limit, how well it would work in terms of genetics.
So we have one researcher in our group that for example, really study youth suicide. And in his work, he finds that constellations of polygenic risks and clinical phenotypes that have to do with stress response, ADHD, impulsivity, are much more important than when we look at individuals later in life, where it looks like constellations of phenotypes that have to do with depression, loneliness, social isolation, maybe the chronic pain issues for some folks are more of an issue. So again, there's kind of a maybe simplistic example of how we're sort of starting to think about this and pulling it apart and not thinking, oh, there's suicidality and it's just one thing, but rather it's many types of suicidality.
And I think that's also important, because at the other, about regarding the question what to do with the information or with risk prediction, because we don't, unfortunately today we don't have any good interventions to offer people to use as a basis. If we have an increased suicide risk, what can we offer? Of course, we have interventions that could address different types of underlying or related conditions, treatment of mental disorders, etc. But there are no specific or very few evidence-based specific methods to prevent suicide, and that's also so. I think in order for risk scores to be clinically useful, we also need better interventions, more interventions, better treatment options, or we have in order for the knowledge to be actionable, we have to have something to offer at the other when we have the information for patients or relatives.
Yeah, I think there's one evidence-based treatment that's being deployed in veterans that has to do really with getting at this impulsive thoughts of suicide and interrupting that. And in that group of individuals who are young men who are, probably have been very exposed to very stressful experiences, that intervention works pretty well. And that's a really good example of potentially one group of individuals at sort of specific underlying genetic risk with particular environmental augmenters of that risk where this intervention works. And it's our task as researchers to sort of try to pull this very complicated problem apart so that we can get at groups of individuals like that, where we understand much better what's going on with the genetic etiology, but then also the environmental stressors that are magnifying that.
If you think of the younger suicide risk subgroup, could you sort of speculate about the trial or an intervention for that, like corresponding to this veterans intervention?
So, I mean, I'm probably not the right person to talk to. I have clinical colleagues that work with these populations. They think a lot about trauma exposure, especially early life trauma, mitigating the rumination about that trauma, trying to pull people out of life situations where the trauma is either perpetuated or magnified. I don't think there's any magic bullet, but I would say a lot of the folks that I know of who treat individuals who are youth or young adults really think a lot about trauma and stress response. So again, that's really almost from their clinical experience, but we can take that clinical experience back into the research and say,
yeah,
how do the genetics match what you are thinking about in your clinical experience? And to a large extent, they are what we might expect. We see this impulsivity, we see the PTSD, the stress response related sort of polygenic risk starting to coalesce in some of these individuals.
One question, do you think that genetic risk scores could help identify who would benefit from these treatments, or? I
love your the glass jars, that's such a great example, because maybe, right? But it may be then that the blue triangles are the things that are really important in putting somebody up to the top of the jar. So maybe, I don't know.
Yeah, there may be genetic differences between the individuals who experience trauma and those who don't experience trauma, differences in biological etiology. It's often difficult to get the data on those kind of environmental risk factors in the large sample sizes that we need to do these kind of studies, given the small genetic effect sizes that we're working with. So that's one challenge that we're facing. But just to come back to the question that just came up about predicting response to treatment, there's not really enough data at the moment to know whether PRS could be useful in predicting response to treatment. Again, it's difficult to get information on treatment response from large populations to do a GWAS of treatment response. So there's definitely work ongoing in that. There's a consortium for lithium genomics. There's consortia looking at the genetics of response to antidepressants, but there's just insufficient data at the moment to know if treatment response could be predicted using PRS.
I guess you could add the therapy genetics regarding psychotherapy as well.
Yeah.
But, we have an interesting question here. We have to, time is almost up. I guess we have time for this one. If the panel and you, Niamh, could develop a bit on the sex discrepancy, that more men actually commit suicide, but more women self-harm. And also maybe geographical differences. I mean, the genetic work is based mostly on Caucasian. What do you think of that?
So yeah, there's major sex differences in the prevalence of suicide and suicide attempt. We would like to be, genetics is just one reason, potential reason for that. You know, there's many possible explanations, but we would definitely like to be able to look at sex differences, and we plan to do that as our GWAS increase in sample size. And then again, in terms of those geographic differences, that's a very interesting question. And again, it just comes back to, we need to further increase the ancestral diversity in our studies and to increase representation for those populations to be able to do sex-specific analyses in those ancestry groups, and then compare genetic etiology between the sexes and between ancestries. So I think that's a great question, and it's a very interesting future direction.
So one thing I just wanted to add to that is that the PGC Suicide Working Group and the leaders, and Niamh in particular, also done a really good job in bringing in collaborators from people around the world where there will be deep knowledge of societal pressures, environmental exposures. So that's also going to be critical. And it may be that some of that is really driving the differences that we see across cultures, and that the underlying genetics is not as dissimilar as we think. So we need to make sure that we always keep both of these pieces in mind, because they're both really important. And that is a huge aspect of this big international consortium, is making sure that we pay attention to the other side of the fence, not just the genetics.
Thanks. Great discussion, I think. And so nice to meet in this context. The time is up, so I guess we'll have to call it a day today and now. Thanks everyone who participated, and thanks to the audience and the organizers and the panelists and me. There will be an evaluation link sent out via chat, which we would be grateful if you could respond by clicking on the link. Will also be sent out via email for those who don't have time to respond now. There's also the next coming seminar in this series is with Craig Bryan, who wrote a book on rethinking suicide. It's a very interesting book, recommend it. So that's on January the 25th, at the same time. So yeah, with that, we call it a day, and thank you very much all.